share my screen. Okay. All right. I'm hoping you guys can see just the slides. Perfect. Awesome. I am gonna try and keep this short and sweet. In the spirit of there's other things to talk about, and leaving time for questions and sort of discussion. But thank you, Embry, for inviting me to give a little bit of information about what I've been working on for the past 5 years, which seems all-consuming. For those who didn't get the introduction, my name's Kelsey. I'm finishing my PhD right now at the University of Victoria in BC, so British Columbia, Canada. But I am co-supervised and have spent much of my time actually in Switzerland at AOVAG, with the Swiss Federal Institute for Aquatic Science and Technology. Both of my co-supervisors, so Dr. Doria and Dr. Stronde, send the regards. They actually wanted to be here today, but they're both on vacation, which is lovely for them. So I am here instead, talking about a portion of my PhD research. What I'm going to be talking about today is sort of the microbiology side of my research, which is really understanding the anaerobic degradation mechanisms that occur in septic and holding tanks or cesspits, in sort of varied geographical settings, which I think there is a lot of overlap between what this group is interested in. However, as we all are aware, septic and holding tanks are not mini anaerobic digesters, and do have a few key differences. And that was kind of one of the um, motivations behind this work, is trying to figure out what those differences are, and what that means in terms of treatment as well as in terms of greenhouse gas mitigation. So before I start, I think it's important to acknowledge where I am today. So I'm currently in Victoria, and I acknowledge and respect the Lekwungen speaking peoples on whose traditional territory the University of Victoria stands, and the Songhees and Esquimalt and Wasanich peoples whose historical relationships with the land continue to this day. And a little bit more geography. So just to give a little bit of context of what this project is and was, and where I was working, and what septic tanks and holding tanks I was looking at. So I'm partnered with, like I said, the University of Victoria and AOVAG, so in Canada and in Switzerland. But another key project partner was the University of Makerere in Kampala, Uganda, where we have a close relationship with the civil engineering department at the university. And specifically, this pertained to the two different field sites where I collected data, and collected my samples from septic tanks and holding tanks. So most of my, half of my samples, I should say, were from southern Vancouver Island, where the University of Victoria is. So this is rural context, with septic tanks. A lot of the southern Gulf Islands are all septic tank systems. And then also in informal urban settlements in Kampala, Uganda, I collected samples from what are called septic tanks, but are actually more holding tanks and cesspits, in the sense that they're not connected to a land-based treatment system. They're just the tanks themselves, typically, sometimes connected to a soak pit, but with different levels of sort of offsite or post-containment treatment. So a little bit of background of why I'm looking at septic tanks and holding tanks, and why I care. So as we know, humans, people, we produce a ton of shit every year. So 360 billion cubic meters of wastewater produced globally every year. More than half the world's population uses on-site sanitation rather than sewered systems. So they're very important, they're very prevalent. And actually, septic tank use itself has more than doubled since 2000, the year 2000. And comparing that to other on-site sanitation systems, so like pit latrines or sort of containment-based systems, like a honey bucket or composting toilets, they have only increased by a third. So the use of septic tanks is increasing um, as more of the population is urbanizing. Oh, I see a question.
Yeah, sorry, Kelsey. So
Yeah,
we just going back to what you had said previously, because and I hear you put septic tanks in quotes. So
yeah,
could you again maybe define for because define for us what you call a septic tank and maybe what you're calling a holding tank? Could you just give us a little bit more granularity on the distinctions that you're making there?
absolutely. That's a fantastic question. So a septic tank, as it's deemed in sort of an infrastructure definition as well as a treatment definition. A septic tank is a multi-chambered, usually precast or fully lined underground containment that collects wastewater at the point that it is generated and is connected to some type of post collection system, whether that is land-based, like in a leach field. Typically, we see the most in North American contexts, or it can be connected to some type of modified sewer network sometimes, or some type of soak pit. So where that liquid portion is being transported and treated somewhere, and then the solid portion remains in the chambered tank itself. The major difference between using the term septic tank versus a holding tank or a cesspit really comes from this sort of treatment side of things. So a holding tank or a cesspit is essentially just a short-term storage vessel where that wastewater that is being generated on site is being stored in a containment and typically is then emptied, both the liquid and the solid portion, without
Mm-hmm,
that liquid
mm-hmm.
portion having any more treatment. So that's the kind of the major difference, I would say,
Okay,
especially in the context of what I saw in Canada versus what I saw in the settlements where I was sampling in Uganda.
okay,
Yeah,
okay, okay. Thank you.
yeah. Essentially, they're just, yeah, big tanks with poop in both cases, but slightly different configurations.
Yeah, and also you get supernatant off of a septic tank, but you don't necessarily. I mean, I guess you do have settling in a cesspit, but it's not as engineered, I
Exactly.
guess. Yeah.
Exactly. Yeah. Which actually leads really nicely into kind of what was the motivation behind this work and why I looked at these systems in general. And that is that for on-site sanitation that sort of fall into this septic tank category, whether that's a multi-chamber tank or a holding tank or a cesspit. They're commonly assumed to rely on anaerobic degradation, and they're commonly assumed to be treating the wastewater that's in situ to some degree before it is hauled off-site. This is the assumption, but the links between what's actually being degraded in situ and the microbes that are there and present in doing this degradation that's assumed, as well as the amount of end product that is produced, so carbon dioxide, methane, nitrous oxide to some degree. These links are still missing when it comes to on-site sanitation, and a lot of the principles from conventional wastewater treatment have been sort of um, applied to these systems without actually verifying if that's truly the case. And so my question was really to figure out what is actually being degraded in situ and who I should say, of the microbial community is responsible for that degradation. And the reason,
Kelsey,
yeah,
Kelsey, can I ask another question?
yeah, of course.
Because when we talk about treatment, are we talking about volatile solids reduction? Are we talking about pathogen reduction? What do we what is our metric for treatment?
Really good question. And perhaps treatment is a bit of a misleading term that I'm using, because in this sense, I'm really talking about the conversion of readily degradable organics into methane and CO2.
Cool.
Yeah,
Thanks.
my work really didn't focus on pathogen reduction at all. However, as we know, that is very important when it comes to treatment. So yeah, I'm specifically concerned with what is happening in terms of the readily biodegradable organic portion that's being converted in these systems. And yeah, I think why that's important for septic tanks and holding tanks and cesspits is it has a huge impact on estimating the quantities and the qualities of the wastewater that is then going to downstream treatment. So you need to know what's actually happening in situ to be able to predict what you're going to be transporting downstream. And there is, of course, the really important question in terms of climate mitigation and how much greenhouse gases are being produced by these systems. And is there a way that we can accurately estimate and predict that based on characteristics of these systems? So that's kind of the motivation behind my work. Um, so big picture, that's what motivated me, but specifically what I wanted to share with you folks today is kind of the really zoomed in view of what in situ microbial ecology looked like in these septic and holding tanks, um, and what the community composition was like, as well as the metabolic potential as it relates to methane production. So it was a really unique study in the sense that I could compare um, geographically very different systems, but ones in which they receive very similar wastewater that is generated. And I wanted to see are these assumptions about their differences in anaerobic degradation, do they hold true? Are they similar to conventional wastewater treatment principles? And what are we actually seeing from a functional potential point of view? Um, so what I did is surveyed and sampled 22 septic tanks in Canada, 19 in so in southern coastal BC in Canada, and then 19 in Kampala in Uganda. I measured a whole suite of physiochemical parameters as well as actually measured methane that was being produced in situ from these tanks, these containments.
Mm.
So measured this in situ at different levels in the containments themselves. So I took samples from the top layer, the middle layer, and the bottom layer. And then as well brought samples back to the lab for more physiochemical analysis. Question. Yeah.
Sorry.
Yeah. No, that's okay.
I get so excited. So I have two questions.
Yeah.
What did you ask in your survey? And like, I'm curious how that informed anything, if at all, or if it was just part of like the background data, because we do have a socioeconomic component in this project. And then you took dissolved methane samples in the liquid.
Two very good questions. I'll answer your second question first. No, I did not measure dissolved methane, and that was definitely a 2020 hindsight moment where I wish I had of. Unfortunately, I only measured the atmospheric component with a flux chamber like over the liquid surface. But not recent and recent research has indicated that there's likely a large portion of methane that is dissolved in the wastewater that is not accounted for in those flux measurements, which is very interesting. But I did not look at it,
But
or I
they
did not
will be
measure it.
when you start pumping.
Yeah,
That's the,
exactly.
yeah, they'll yeah.
Yes, and then from the survey point of view, so the survey that I used was mostly geared towards like construction and operation characteristics. So what type of wastewater was being generated? Was it just black water, so just toilets? Was it also kitchen, bathing, laundry water?
Mhm.
When was it constructed? How often do you empty your containment? When was it last emptied?
Mhm.
So no sort of like demographic data was
Mhm.
collected in terms of like income level of the household or personal details like that. It was mostly related to construction and operation itself.
Mhm.
Yeah.
I have more questions, but I'll shut up. Keep going.
No, no, I like questions. This is great. So yeah,
Or put them in the chat,
yeah,
throw them in there just so you don't forget them.
true. Um, so yeah, I measured all these physical chemical characteristics. But importantly, what I also did was collected samples in situ for further amplicon and shotgun metagenomic analysis to really dig into what is the composition of the microbial community, what is the metabolic functional potential of that microbial community, and how is that relating to the physiochemical characteristics that I'm seeing, as well as the methane production that I'm seeing. So that was a big portion of this work. And I wanted to throw in some photos of field work because I think that's always fun and exciting. But this is just some examples of what that looked like. So the two septic tanks on the left and right, these are from two containments in BC, so on the southern island here in Victoria or around Victoria. The one in the middle is a containment in Kampala that I sampled from. And the little orange PVC pipe is my um, designed flux chamber that I used to capture that headspace gas from a sampling port. And that's what you can see me doing in the middle there is collecting those gas samples, as well as some probes that I use to measure dissolved oxygen, electrical conductivity, and pH in situ. And again, more photos. I have lots of photos. Um, just collecting different samples at different depths. So that photo on the left is a grab sampler that I use to be able to let me take samples at the bottom, the middle, and the top without having other layers get into that sample. So trying to contain it to compare those differences. And then using probes to measure in situ and lots of lab work afterwards, which was a very big part of my life for a very long time. And now the actual findings and what I think is maybe interesting for us in this group is what did I actually see in terms of the microbial community composition, and how did that relate to the functional potential? So, from all of these containments, I did find, and this is from the amplicon sequencing of the DNA that I collected from samples in situ. It revealed that the same three phyla accounted for more than 86 and 76% of the total relative abundance um, of the microbial communities in southern coastal BC and Kampala, respectively. And I also compared this at the family level to other human waste environments. So I compared it to um, data that we have from the human gut, from the Human Microbiome Project. I compared it to other studies that looked at containments as well as pit latrines in Lusaka in Zambia. And I also compared it to the Midas, which is this big wastewater microbial community wastewater database that has been conglomerated to see what the similarities and differences were. Obviously, this is at the family level and it's only composition alone. It says nothing about actual function or activity. But what we do see is that these septic and holding tanks that I sampled from have a very characteristic microbial community composition, which is relatively distinct from other human waste streams. So these tanks themselves are they have influence from our gut, and they have influences that are similar to conventional wastewater treatment, but to themselves, they are rather unique and they're a unique environment, um, which is very interesting. And I think when I start talking about functional potential and treatment mechanisms, or I should say degradation mechanisms, that's very important, I think, to remember. And then, like I mentioned, and again, all of this is unpublished. I'm currently trying to wildly write up these manuscripts to defend. So this is preliminary information. Um, but the functional potential redundancy, so I looked not only at the composition, but with the shotgun metagenomics, the potential energy metabolisms of these communities. the functional potential was very similar across the containments themselves in both locations and between individual containments. So there was a lot of functional redundancy despite taxonomic variability. Um, and I'm realizing I'm not reading the questions as they're coming in. So I'm sorry if I'm missing anything.
I didn't. I did that to so we could wrap up
Okay.
at the end.
Okay, perfect. I will just continue then.
But I've monopolized the question. So Alex, if you want to jump in with your question.
No, I think that's fine. Let's take questions at the end and keep
Okay.
keep ripping.
Okay. Um, ooh. And then in terms of looking at the differences in terms, because I did measure at different depths, and I took samples at the different depths as well. And I also collected all of this physiochemical data on different properties of the wastewater and characteristics of the wastewater. And what I found is that we did see distinct compositions. So again, making sure that I'm being diligent in saying that the composition, so the actual community members of the containments, we did see distinct compositions between BC and Kampala, but the functional potential when it came to the actual energy metabolism of these communities was not very different. There was a lot of overlap and a lot of redundancy. Um, and even at the composition level itself, we didn't actually see much difference between the different depths in containment. And this was kind of an assumption that was surprising to us. We thought there would be more distinct differences at the community composition level between top, middle, and bottom due to settling, due to different degradation processes potentially happening. But it turns out that what enters the containment. So in terms of what type of wastewater is generated and how it's entering the containment could potentially be more important in the characteristic of the wastewater and the community than what happens in storage during storage in the containment itself. And I could talk about the nuances of these results for a long time, but in the sake of time, I will continue on. And something else that was really interesting is that from the shotgun metagenomic data, we realized that methanogenic genes, so the genes involved with the last step of methanogenesis, so producing methane, they were consistently abundant across all containments and all of the samples that we collected. But the measured emissions that I collected, so the flux chamber and all of those point source gas emissions did not seem to correlate at all. So there was a lot of variability in the point source emissions that was not reflected in the consistency that we were seeing in those methanogenic genes being present. And we saw both acetoclastic and hygienotrophic methanogenesis genes present in the majority of those samples. And so this was a very interesting um, result because it leads us to believe, and we did more, um, there were more experiments after the fact to dig into this more, which I won't talk about today, but it kind of leads us to believe that the methane generation in these systems is rather activity limited than community limited. So just because certain community members were present did not necessarily mean that more methane was being generated. Also, on top of that, it kind of led us to reevaluate if flux measurements themselves are actually representative of the methane that's being generated in these systems. Um, so yeah, that's kind of the okay, now what or so what, which is, now we can move forward from asking who is present in the containment, but under what conditions they perform and how that performance can be measured accurately. So I had more slides, but I think I'll leave it there because I think that's a lot of information. And I will move to questions. which I'm sure we have a lot.
Yes. Now, Alex, why don't you go?
Take what you want and leave the rest. You don't have to take my question. I'm actually, because we're working on this um, NATO project and Jane is leading that, I just assume you take her questions.
I will, let me look at the chat here. Jane's question. Okay, photosynthesis is substantial in your functional potential. Since this is likely not actually occurring in the system, what do you think this signal is related to? Yeah. Um, oh, and also the methanotroph question. Super good questions, and also something where I was very surprised um, to see the photosynthesis sort of signal as well. So for this work, because these were samples that we perform shotgun metagenomics on, which is just a measure of that functional potential and not activity itself, is likely just for photosynthesis is just a shadow of basically like plant material or especially when it comes to sort of vegetation and vegetables that we're eating in our gut being present in the containments themselves and not actually that the photosynthesis processes were abundantly active. So that's kind of the downside of just doing shotgun metagenomics and not pairing that with metatranscriptomics or stable isotope probing, for example, which is what we did in our future work. The results I didn't show was to actually look at activity. So that's what we assumed was responsible for that signal, I should say. And then as for methanotrophs, we did not see any strong signals of methanotrophs in these systems, which was also somewhat surprising because I was expecting there to be some present. Whether, it just wasn't detected in the samples due to degradation of the samples themselves or the systems that we sampled and them not being present, I can't really comment on that, but we didn't see any signals of those in our systems. Alex, did you want to ask your question?
I just didn't understand that overlapping graph. So, you look at
Yeah.
different substrates that you're going to put into something, a feedstock, whatever the heck it is. What is overlapping with what there at those percentages? I don't understand that graph.
Yeah, I can maybe just go back to that.
I mean, it seems promising because more overlap is kind of good, I guess.
Which one are you? This
Right?
one?
I think that one, the overlapping.
Yeah, so this, and I spoke about it very quickly. But essentially what this was a comparison of, or what this showed, was I took the community, so the microbial community composition results at the family level from all of the containments that we sampled, and then compared that to, at the family level, the databases that are available from the Human Microbiome Project and the database that's available through MIDAS, which is this global sort of wastewater, sewered wastewater.
So 48% of the um, family level things that you saw in your field work in Lusaka, Kampala, wherever, were also in what?
Oh, okay. Yes, I see where the confusion is. So the orange bar, that 48%, that is the amount of overlap with a separate study that looked at containments and pits to my results, to my containments. And then I only saw, so that was the highest amount of overlap was between other containments and pits and my containments. Not mine, but I'm using mine. Um, folks who I sampled from. And then there was only 24 and 37% overlap between the contaminants that I sampled and the human gut and sewered wastewater databases.
Okay, so the orange is yours compared to a different study, and the
Correct.
green and the blue are compared to the orange.
Correct. Yeah.
I
And
got it. Okay.
I probably chose the wrong figure to put here because I have a Venn diagram that better explains the relationship. But this was the only one that I had ready in my R results for today's presentation.
Thanks. I got it. No, I just didn't understand what was compared to what.
Yeah.
And if I don't understand, I would have to sing a song that is by Les McCann, which has some vulgar language, but the song is called "Compared to What."
Okay.
Anyhow,
Quick, quick, find something that Alex has to sing a song about.
well, I missed our multilingual national anthems. Sorry.
Do you have that?
Oh, Canada.
You know the French one?
I don't know that one.
You're muted, Alex. You clicked something.
I can hum it. I don't know the lyrics.
The Canadian one or the French one?
The French one.
Oh, I meant the French version of the Canadian one. So,
Okay. Yeah. Because the French one is a very bloody one. But,
hmm.
Alex, you're still muted if you're trying to sing. It's not working.
Sorry.
Purposely.
No, I was going to say something about the agenda. Let's. What else
Oh,
on this?
wait, I have one more.
Yeah, go ahead. Keep going. Yeah.
One more question, please. So at the beginning of your presentation, you mentioned that there's going to be like sometimes we think of septic tanks as overlapping or a kind of like a proxy for some anaerobic digestion characteristics. So what are the implications for your findings vis-a-vis like a small anaerobic digester? Like what are the right? Because in my mind, like the anaerobic digester that we're looking at, we're gonna dump some poop in a tank, right?
Yeah.
So what do you think? What do you think the implications or the distinctions or the variations between your results and our kind of pilot rural digester might be?
Yeah, that's a really good question. I think one of the biggest things or results to come out of this work was really just like how stark the difference is between these systems and like engineered anaerobic digesters in the sense that and maybe I'm like preaching to the choir on this one, but septic tanks and holding tanks and cesspits are not anaerobic digesters and their communities are different and the level of degradation that is happening is actually much less than I think people assume is happening. Um, and so in terms of that overlap, obviously for this project, we're not saying we're going to build a septic tank and assume it's going to work like an anaerobic digester. So there are those engineered components that septic tanks and holding tanks don't have that we will have in this project. But I think one of the biggest findings in this work was that I think there is this assumption or this idea that the homogenization and the degradation during storage can really change and shape the microbial communities and shape their degradation processes. And, to some result, I think, or to some extent, I should say, I think that is happening. But really, in these systems, a lot of the differences, it's funny. So a lot of the compositional differences we were seeing seem to be coming before any storage in the containment itself, and that had less of an impact on the composition, and yet the functional potential was quite redundant. And so I think these systems have a lot of sort of like functional guild interaction when it comes to how they're actually converting these organics into methane. But what is coming into containment is really important. And what's coming before storage and containment is really important in terms of shaping that environment. And I think, there is some overlap between that idea and what is being talked about for the small digesters in community, obviously with major differences in terms of operation and adjusting for parameters. But I think that could be an important sort of learning. That's, yeah,
Would it
my
cost
opinion.
less than or more than $1 million to have you do this same work for small digesters? I'm just kidding, but I guess what part of it, if I want to
that was
boil this
going to
down
be
and be
my.
really, we're really coarse about this, is like you're saying that maybe the amount of degradation that's happening in these cesspits is not what people think, and it's not as good as much as what people think. Now, on the one hand, you can say, "Oh, great, that means our digester project is going to be very nice in comparison," but we don't really know how effective our digester stuff is going to be in an apples to apples kind of way by using your same methods. So I'd love to again make some wonderful claim about how wonderful this digester is going to be compared to current practices, but I guess we can't really say that until we know how this digester is going to do on the same kind of parameters that you looked at.
I mean, and I think like Jane is definitely the expert in this. So please jump in if I speak incorrectly. But I think the benefit to what these digesters will be compared to like a septic tank or a cesspit that I was looking at is like I had no way to measure the loading of organics coming into the system. Like
Mm.
I didn't know how much wastewater was being generated every day. I didn't know because septic tanks and cesspits, it's this mix of partially degraded and fresh wastewater coming in all the time, which of course you have in a digester as well, but you have more ways to like quantify that. Where I was coming in and taking point source measurements and making assumptions about the loading, but not actually knowing. Um, and there is this, you know, there there are these black boxes essentially in the ground, and the amount of BOD and COD reduction that you're seeing in a septic tank or a cesspit is usually less than 30%, which in itself is telling you that there's not a lot of degradation happening there. Whereas, in a mini digester, you're looking for more than that typically. Um, and you can have ways to actually measure the influent versus the effluent, whereas I didn't, and then I'm trying to sort of fill in the gaps, so to speak. So that's a benefit compared to these.
Yeah, it would definitely be useful to talk about sort of, what your lessons learned. Like you said, you should have measured the dissolved methane and you didn't, and other things that might be helpful for
Freaking
us
hard.
in this project too. Um, yeah, because I think we're planning on sort of taking a similar approach, like doing metagenomic analysis, and then we've talked about either metatranscriptomics or stable isotope probing on this
Yeah.
digester to get what's going on. Yeah.
Yeah. And that's unfortunately those are the results I didn't present, which is I took samples from a couple of tanks and then I did qPCR and metatranscriptomics work with those samples in microcosms, though, so like in lab controlled microcosms to look at the proportion of the methane that was being generated by acetoclastic and hydrogenotrophic methanogens, as well as the impact of like substrate inhibition, et cetera, which then kind of was able to answer more questions related to that activity piece that I couldn't really answer with this work. But of course, I didn't do that in the field. I just did that in the
Well, I
lab.
think that's not really practical to do in the field, at least just based on the costs of like 13C labeled substrates. It's just, we
Exactly.
would do it in the lab as well.
Yeah. Um, yeah. So, I mean, that that would be if I hadn't have done that, that would have been like my recommendation to myself is to look at that activity piece of it. But I think I was again quite limited because I could only bring two samples back from two different containments, which didn't tell me much about like the greater data set. Whereas like potentially the nice thing with the digesters that you eventually implement is you'll have them for long term. You'll be able to take multiple samples at different time points under different operating conditions, which is kind of what you want, right? So yeah, it's
Well,
exciting.
I think we'd love to have you back and hear about your stable isotope probing and transcriptomic results too. That would be
Would
cool.
love it. And hopefully after my dissertation defense, which should be very soon, I will have lots more to say.
Yeah, congrats on that.
Well, with all these questions, I thought this was your defense.
I wish.
Or we're like your SAT practice session.
Yeah, very true. Yeah, practice is definitely good for me at this point.
So I have a question for Jane, actually, because I think we've been talking to the biogas guys about seeding the digester with lagoon, an anaerobic lagoon seed, right? And now I'm just curious if there's any implications for the microbial community that we are sourcing for the seed, and if that's going to impact like startup and stabilization and steady state, because it's a different, potentially from an anaerobic lagoon, which again in my mind is a kind of like a septic tank, but maybe I shouldn't make that presumption. So are there any microbial community considerations for our seed that for the digester that we want to sample or track or quantify or characterize?
Yeah, I think that's a good question. It would probably be possible. So we just kind of had in mind that the lagoon, like if you take a deep sample from the lagoon, it's going to contain a lot of the types of organisms that you want to have there, like centrophic fermenters and different types of methanogens, and that this would probably work as a good seed. But we can do some like characterizing, just like simple characterizing of the community, just to see like who is there, to see if the organisms we think we want are there. I think generally when people start up digesters, and of course, like I think the most important thing is to have something like that's adapted to the sort of climatic conditions, because these organisms seem to be in a lot of places, and they will kind of grow from either the feedstocks or the inoculum that you provide. And it might be a bit slower startup than it would be in like a mesophilic or thermophilic digester, but I think that we'll find the right organisms somewhere and get them to grow.
Yeah.
Just give them what they want.
Yeah, food. Thank you, Kelsey. It's fascinating. I love this stuff. Thank you.
Thanks. And yeah, if there's more questions, I think my email is available, so I'm happy to answer more questions too.
All right, Embray, Margaret, what are we doing? I put that agenda up there, but I kind of wanted to, I guess the other big thing is to hear from Jane and um Jaime is here, I guess, and there was some design meetings. I was in one of them. Embray, you had to go take care of a tree and a power line and all that stuff. I guess we could touch on that too, but uh, whoever has some updates on that or things to share, please take the floor.
So I don't have too many updates about the NATO project right now. So we're just about to submit the first technical and financial report at the end of this week. So that's kind of been taking up a lot of the time. I am meeting with my procurement person at SFU today to start discussing how to set up the contract between Balanced Biogas and SFU. And I have talked to our lawyer, and he finally agrees with me about that we're going to have a service contract and a research agreement. So finally, we're on the same page about that. So baby steps on the progress towards that, but I think it's at least headed in the right direction. Yeah, and then I don't know if Jaime can comment on the design. Unfortunately, yeah, he didn't make it to the design meeting last 2 weeks ago, I guess, but maybe he can make it tomorrow. So, Jaime, do you have anything to add about the design?
Hello, nice to see you. Well, I load some archives into our Drive page, so I don't know if you see it, but I can talk about the design, what we are discussing with Davide and Alex, if you want. I don't know, well, this is a simple design. You are thinking about with a digester of 55 gallons with manual feeding and with a download by overflow. So let me show the slide. Maybe if you have some questions, I'll try to answer them. I have put some other things in the design in order to, well, let me show you. Can I change? Do you see my screen?
Yep.
Okay, this is the design of the digester. This is a PVC digester. We can build it with HPDE, but I think it's a solid shape is much better in order to cover this with a heating system. It is possible. And we suggest to put 2 axial sample ports in order to take some samples to measure, for example, what Kelsey measured, the content of make some qPCR analysis in the samples, how if maybe we have some stratification of the microorganisms families along the digester. So we can measure so many things. And we have here a thermometer into the digester. We can measure the temperature, or we should measure the temperature inside the shed. This is the shed, and the outside of the environmental temperature. The idea is to make a kind of a model to see the interaction of the temperature and how it can affect the microorganism process. Also, the idea is to cover the digester in order to increase the temperature and reach temperatures above 10 Celsius degrees. Because we are now measuring in the laboratory here in Colombia the methanogenic activity above 10 Celsius. So if we cannot reach this temperature, it's really difficult to make methane. So this is the idea of the design, covering the digester with the shed, and maybe there is a possibility to put the digester into the building, as I understand. So, well, this is a simple design. We want to build it here to have in order everything what we are gonna need to install it in Yukon. So the idea is to build it here in Colombia, make some proofs, and then list all the requirements to build it there. Well, I don't know. What do you think?
I'm going to put something up that was my notes for this. No, I'll just say them. 2 things that came up when we were looking at this slide was that my brain was always thinking bigger, right? And so why so small? But it has to be in this building if that's what we're saying. Therefore, it's 55 gallons. We also said that it could theoretically be a bag. So that was kind of interesting to me. As much detail as this had, I could picture it having the same components, but in a bag type system. We talked about feeding. I asked about why a 10-day hydraulic retention time. And you said something about kinetics, and I don't know what that means by kinetics. And you also said maybe could install, or we said, somebody said could install 2 digesters for comparison, 20% maximum solids, And then what will we use for substrate? Those were a bunch of things that we talked about in addition to what's mentioned here, or talked about when we were looking at this slide.
Yes, well, these designs also allow us to build maybe a battery of digesters. We can put 2, 3, 4 digesters in parallel, yes, in order to increase the substrate or the waste that we are gonna treat. Well, we can build it in a bag too. Yes, it is possible, but I think it's more difficult to operate. Yes, because the shape, we cannot keep the shape stable. So it is going to get inflated, it is going to lose pressure. So maybe the dynamics is going to be different. Also, actually, we always, when we build a digester in plastic, we put it into a trench, but we cannot make a trench there.
Yeah.
So we have to make like maybe a system that allows us to keep the shape stable of this bag in order to avoid altering the dynamics of the fluid into the digester. So there are so many possibilities. As a substrate, we have here an example. This is a PVC digester where we have so many of these in our laboratory in order to prove some different substrates or inoculums. This is a 8 liters digester. The students or anyone who wants to play with this toy can feed it manually, and they can remove the digestate by gravity, yes, by overflow. And as I told you before, this design allows us to put a blanket in order to increase the temperature if it is necessary. The limit would be the temperature that can allow the PVC pipe. So it's more or less 750 Celsius degrees, okay? And I don't know, if you see there is 2 axial ports, I play with this and I take some samples every week and I prove the methanogenic activity along the digester. And the first sample port, the methanogenic activity is low, but in the second is higher because we have a zone where the microorganisms produce the volatile fatty acids, and the second part of the digester, they consume it. So there is the methanogens. So this is the importance that why we are proposing a tubular digester.
Honey, what % solids are you feeding these digesters, your lab digesters?
Well, it depends on the substrate. Mainly, we use manure, for example, cattle manure or pig manure. We have some dilution ratio that we have established as most favorable. For example, for cattle manure, we use 1 part of cattle manure and 3 parts of water. So the substrate is around 50 grams per kilogram of volatile. Between 50 and 80 grams per kilogram of volatile. So we always dilute the wastes because of the dynamics of the feeding with this kind of digestion. So we cannot make anaerobic digestion of solid wastes. It's necessary to dilute. And as a substrate proposed for the NATO program is, for example, food waste. But it is necessary to make a pre-treatment of the food waste in order to reduce the particle size and make some dilutions in order to make that fluid into the digester.
So let me see if I understood that. You're asking % solids, Embre, and I had jotted down during the meeting 20%, but 50 to 80 per grams per kilogram is way lower. So are these being operated, at a much more dilute or much lower solids fraction?
Yeah. Yeah, because when you said 20%, that made me nervous.
Okay. Well, maybe I got that wrong.
No. I'm not saying you got it wrong, but I was like, how are we getting our solids percent down to 20%? Because that's a mechanical exercise, and then it seems like it would be a mixing issue.
Yeah, and to that question, maybe this is something people already know, but in the earlier diagram, the length to diameter ratio was 3. These look like they are way higher than that, and that probably has to do with the movement of the material or the percent solids. I don't know. Sorry, I'm just noticing that too.
Yeah. So Jaime, on this question, going back to your design, the design where the length to width ratio is 3, what percent solids do you think would be optimum for that configuration or that geometry?
Okay, well, I would prefer with this dilution. We also have proof food waste.
Yep.
But I don't remember the dilution. But it could have reached more of 80 grams per kilogram of volatile solid.
Okay.
But it's really difficult to operate to feed the digester. Yes.
Yeah, at higher percentages.
Yes, because of the flux. Yes.
Yeah.
And this digester that we have here has a length diameter ratio around 7. Its length is higher because we want to simulate a rural digester that has this ratio is longer. But because of the consideration of the building that we want to put our digester into the shed, so it was necessary to reduce the length diameter ratio.
So there's some code issues if we go above around 220 to 250 liters. So that was part of the sizing issues. And then the other issue is if we want to build this outside of the building, the doorway is 1 meter wide. So it has to fit through there.
Okay, Nate, do you have any design input here before you head out?
Previously we were talking about the bag digesters that Dr. Jaime has, and usually those are like in the ground to have a structural support. So I really like this pivot to some sort of rigid or even a bag that has a small support, because ultimately, we need to fit inside the building that we have. So it looks like we're already moving that way. Thank you, Amy.
Jaime, can you make it to the design meeting tomorrow morning?
I don't know. I would like, but actually I'm here because there was a pouring rain in my area of work, so there was the mountain was covering the way, so I couldn't reach my work area.
Oh no. Is everybody okay?
Yes.
Okay.
So I'll try, but maybe I will have problems to travel to my work tomorrow, so I can join the meeting tomorrow.
Okay. But maybe I saw the document you added to the drive. Is it okay if I send that to everybody on the design team?
Yes.
Jane, would you do me a favor and link that drive to this chat and see if people can access it? Because I was struggling a little bit. I just want to make sure everybody can get into there.
Yeah, so you should have access to it, Embry. I don't think that everybody has access to it, but you definitely have added you to the drive.
Oh, I remember what it was. I couldn't upload. I don't have upload rights. I have
You
access.
should be able to, but you still can't because I double
No.
checked it and it said you should be able to. I can check again. I'm not sure. Um,
Thanks for putting that link in the chat.
Yes, I upload a document where we resume those slides for your review. So we propose, for example, a substrate food waste. We also could use as a substrate a mixture of standards. For example, well, the flux here into the digester is considerably low. So we could make a model substrate with a mixture of one carbohydrate, a lipid, and a protein. Well, it is under your consideration.
Sure.
But I think it would be more expensive because, well, here in the laboratory, as for example, as carbohydrate, we use cellulose. But when we, in a period of time that we didn't have the standard of cellulose, we use paper.
Yeah.
For example, as a lipid, we could use oil, maybe.
Yeah.
And
we do have we do have the fake poop recipe from Fairbanks.
Yes. Yes.
That
It's another alternative, yes. For example, we could make these model substrates trying to simulate the diet of the people there in Yukon. We make some review of some papers, and the people in other areas consume many proteins.
Yeah.
So this is a start point.
Yeah.
Maybe.
Yeah. And we did talk about that at one point, trying to kind of adjust the feedstock for the diet.
Yeah, if we're able to use sludge then from the
poopy plant.
Yeah, then that would reflect the diet.
Yeah. I think so.
Well, Alex talked about the kinetics. Well, we propose also minimum hydraulic retention time around 10 days, but it depends of the substrate that we are gonna use and the inoculum. And I talk about the kinetics because we can make some batch assays where we can see the behavior of the consumption of this substrate. I don't have here a graphic, but we can build it. The batch kinetics is like this. Okay, we have a lag phase where the microorganisms adapt to the substrate and they start to produce methane in constantly. And when they consume all the substrate, they get into a phase where they don't grow. So the minimal hydraulic retention time is this. This is time, maybe 10 days. But it depends on the temperature. If we increase the temperature, we can reach the faster. Yes, we can reach it faster. And if we operate in 10 Celsius degrees, maybe we are going to need more time to consume all the substrate and increase and feed again the digester. So I don't know if I explain it well.
Perfecto.
Okay,
Perfect.
gracias.
Thank you.
So here, as a parallel step with Karina and Jose, we are trying to adapt an inoculum to 10 Celsius degrees. So we get the inoculum and we increase the temperature to reach 37 Celsius degrees. This is our standard temperature. This is our control. And another part of the sample of this inoculum, we are reducing its temperature until reach 10 Celsius degrees. So we want to compare the methanogenic activity under those conditions and maybe make a model with a surface response methodology to prove what are we going to have, what methanogenic activity can we reach at different temperatures with an adapted inoculum. So therefore the idea of a minimum 10 Celsius degrees. So well, this is like a backup of what we are doing. And the inoculum that we are using now is similar to it was obtained from a septic tank, but its methanogenic activity is considerably low. I don't know, maybe the soap, maybe the legia is affecting the methanogenic activity of this inoculum. And the presence of oxygen. I don't know how, for example, they are operating the tank that Kelsey showed before, for example. Well, there are so many questions, there are so many commentaries, but I don't know. What do you think?
Jane, how practically oriented is this scope of work? Because the more we talk about synthetic this and that, and inoculum has to be this or that, the less it seems to be a robust thing that could be emulated in the field. As an actual waste treatment thing, it's hard to build on theory. It's better to build on something more practical. And I don't know this stuff, the microbiology is not mine, but my impression is that there's all kinds of microbes in any given mixture from anywhere. And so if you put them in with some other feedstock, the ones that are appropriate are going to thrive. And the population of those is going to out compete the other ones, which will maybe still be there in small amounts. So it always seemed, my impression was always that it was pretty flexible. As long as you didn't change the temperature too much, as long as you had enough possible microbes, something was going to happen. So I'm wondering how you're thinking about that. Do you want more control over what is happening in there and what went in, or do you just want it to be like whatever's there will thrive? It just might take longer.
Yeah, I think that's true. I think if you have something that's really well adapted to the system, you might get a faster startup. But ultimately, if you have some kind of anoxic or anaerobic inoculum, you should get like effective startup at some point. And I guess with the temperature being low, the question is, will that take longer and how much longer than like a mesophilic or thermophilic system? So yeah, I mean, I think the idea is to try to set this one up with a inoculum that's local. And I think a lot of the evidence in the literature is that if you start with a cold adapted inoculum, then that's going to work in a cold system. And I think we can kind of start there. And if it doesn't work out the way we think, I guess we need to figure out something else. But I think it's worth trying to do it in a way that could be most flexible.
And
For
so cold adapted inoculum, we're not talking something exotic there, are we? Or is it somewhat exotic to find
Well, do you
that?
think sludge from the White Horse treatment plant is exotic?
No, not
So
at
I
all.
think
I mean,
that's the kind of hopefully thing inoculum that would work, right? And we can, do
I
some
hope
tests
so.
on it ahead of time to see like what the methane potential is. But I think ultimately, I don't know how much time we'll have for that, given the timeline that we want to start up in. So, you know, if we can just throw it in there and see what happens, we can kind of do those experiments alongside and see how the startup actually works. Or is that too like flying by the seat of our pants? I don't know.
Speaking of flying by the seat of it, never, that's not right. You mentioned in passing, you were like, oh, 1 or 2 years. It sounded like maybe this was going to go longer than we were fearing it was going to be able to go.
So I think what I meant was so 1 or 2 summers. So we want to run it this summer and then
Yeah.
we're gonna let it kind of go dormant over the winter time, probably freeze, and then see if it starts up next summer. Or what does it take to start it up next summer?
I owe a little bit of a calculation for the meeting tomorrow. I'm not sure I'll have it, but I think 77 liters of biogas from a family of four or something like that was a number. And then if you can get that amount of biogas, is that enough to keep your digester warm and to thaw the digester? And Margaret's paper was 734 kilowatt hours. So, um, I just want to compare those two numbers and see potentially if there's enough waste and biogas production to thaw a digester, for example. Yeah, just what's there in terms of energy potential versus energy demand.
And I guess the question of that would also be like, at what temperature are you trying to thaw a digester? Because that'll make a big difference as well.
Yeah, it's mostly the phase change, but right, just just the that's where that was what it was. It was just I think that was the 734 was purely
Okay.
turning ice into liquid.
Solid to liquid. Okay.
Yeah.
Could I just come in on the inoculum question? I think you should view a digester a bit like making sourdough bread, in the sense that the inoculum or the starter culture that you use is always better if it's one that's known to work well in your kitchen environment. Um, Even when we're doing really, really big digesters, we would always prefer to use inoculum from another comparable digester, which is doing the same job. And I think this will be true here. What that means is that if you're building a massive, say, a food waste digester, and you don't have inoculum from another digestion tank which is handling food waste, and you use slurry or something like that, or manures as your inoculum source, it will take you up to, even on a big, relatively warm digestion, top end mesophilic, lower end thermophilic, you'll lose potentially 3 months of startup time on a big digester if you don't use a comparable inoculum. And I think that's going to get longer and longer the closer you are to low temperature systems. So to cut a long story short, I think part of this maybe needs to be to find how to keep the inoculum over potentially over winter, like your sourdough starter and stuff like that. So yeah, keep it as close to what you want to do as possible. Hope that makes sense.
That gets a light bulb. That doesn't just make sense. That's like, thank you.
Okay, I'm sorry, I'm going to have to shoot at the top of the hour here.
Oh,
So
before you
I
go,
was
Angie chimed in, and I want to just say also for you, Robin, one of the things on the agenda is can you tell us what the heck these Scotland meetings are? And Angela, I wanted to hear about your victory lap in North America, and who you're going to be staying with, and where you're going to be breaking bottles of champagne, and what ships are going to be named after you, and stuff like that. So first, Scotland events. What's happening with that? And then Angie.
Okay, well, as far as I'm aware, there is only one meeting that of particular interest to the group, and that is the wastewater very big conference event in the autumn in Glasgow. And Alex, I did send the
Yes, I think I remember now.
the link for that. So I'll I don't know if I can get it.
No, I'll find it. Thank you.
Yeah.
I'll put
Okay.
it.
I think it appeared as if there were two separate meetings, but I'm not aware of two, unless again I'm making a mistake. But I think there's just the one. But it is a big event. We have put in as a team an application to run one of the workshop sessions, and I haven't heard anything. It wasn't me that actually sent that in. So I don't know, Jane, whether you got anything back from anybody on that, or it may be a question for Embry, to be perfectly honest.
Both of those folks unfortunately have departed, so we won't hear from them right now. Maybe we could hear from Angie?
Yes, sorry, I'm on my phone, so you might be looking at the ceiling here. I can't quite tell, but no, all it was is that I was saying that I probably will be coming to Canada in well, I know I'm there at the beginning of August for a conference, and so I'm going to be trying to stay around in August, September, possibly pushing into October time, depending upon it. So I might be able to, just lend a hand if there's actual hands that need lending, if that's at all helpful. And I still haven't quite decided. Were we deciding, because Robin was talking about the inoculum, which I agree with, just to circle back, but are we talking about using actual sewage sludge in this digester and not a synthetic version?
Well, Jane stepped off the call, but she was saying White Horse sewage treatment plant material. So I think she was saying she was going to use what was readily available. And then that, to me, given what Robin said, means that you have to have a similar feedstock as the treatment plant is using. So that sounds like that's what I inferred the approach was.
Yeah. Okay. So simply because it makes a lot of sense when they're actually feeding it with sewage sludge, and we use sewage sludge in our labs all the time because it's a fairly stable and easy to digest thing. So it makes sense that you inoculate it with that, or even from a sewage sludge digester, which probably would be asking too much, but certainly then feed it with that sewage sludge.
Wastewater lagoon and White Horse. Okay.
Wastewater lagoon. Yeah. So they'll be able to at least get some fairly similar microbes.
Yeah. And again, like as your travel plans are firming up, you can get in touch directly with Jane or whoever, whatever site you want to go to. If I can cook up something fun for you to do in Vermont, I will.
Absolutely.
I actually may have some meetings that really coincide with what you are doing.